Okay, good morning, everyone. Uh, I remember you that today we finished the course of Professor Choi, and we finish about 10 o'clock, so that we have after conference from Professor Wanderland da Silva Bolzani. I see that by have the program, I know that today who closed the Professor Short course is Professor Wanderland, and she gave a lecture to compare and to show you how metabolomics is very, very important in the chemosystem. Okay, then you have the course only to 10 o'clock. And about 11, we have the Professor Vandela conference. Okay? Please join the case.
No need a microphone?
No, actually no.
Thank you for coming. Today I want to continue the leftover of statistical qualitative analysis of tomato. Yesterday, mostly I focused in principal component analysis. I hopefully, I understand, you understand all the things of a basic of a principal component analysis. Most of multivariate data analysis is really similar to principal component analysis. The second level, we call it supervised method. We need another data set like this one. Yeah, this one, for example, is chemistry data, like a metabolomics data, NMR, mass spectrum, or UV spectrum. What if you have bioactivity data or classification data, then you have to correlate this one. As you mentioned, unsupervised method is to target maximum variation. So in this classroom, the maximum variation to discriminate the people is a gender, man and woman. So whatever you do, PCA, always PC1 or PC2, the major PC, the first pieces shows the separation between man and woman. But our goal, sometimes our goal or our wish separation is not maximum variation. Sometimes it's hidden, which is more important for your research question. In that case, you can add a second data set, Y data set. X data set is metabolomic. So here, good. And chemistry, usually when you are doing metabolomics, the first data set, basic data set, X data set, is a chemistry data. Then it should be very quantitative. It's very quantitative, like intensity, 0 to some unlimited, very high signal and those. But you have to be very careful about some chemistry data, even carbon-13 animal. Carbon-13 animal cases, actually, if you are using broadband decoupling, it's not very much the proportional. It's not quantitative. Quantitative. In that case, maybe you can give a multi-level, small, medium, and big, something like that one. You can put it there. But usually, chemistry data is for quantitative data. You don't have to see. But why they say is very different. For example, now you want to find which metabolite has anti-cancer activity. Then chemistry data of all your plant material, Y data set, for example, the inhibition, enzyme inhibition data. Then, simply speaking, PLS or the supervised method, you have to correlate which signal relate to anti-cancer activity. But before this one, yesterday, I mentioned, you have to understand the character of Y data. Now, simply speaking, if it's an enzyme inhibition, 0 to 100%, right? Then you can put it there, 1%, 99%, 79%. But you have to understand biological data usually not sensitive as much as chemistry data. For example, 80% of inhibition and 81% of inhibition. Is it really 1% is higher, 81%? No. Then you can transform. Easy way is active and non-active. Active is 0. Non-active is 0. Active is 1. Right? Then you can put 1. We call it is a dummy. One classification. Otherwise, you can put multi-level, active, non-active, or in between, middle active, slightly active. You can put 1, 2, 3 level. Then you have to consider also the character. If you put 1, 2, 3, are they related or not? For example, 3 means, is it 3 times higher than 1 or not? That is a related one. But if I put Brazilian classification, orange is 1. Korean classification is 2. German classification is 3. Then if you put 1, 2, 3, is nothing with not at all of the not at all of quantification. In that case, we call it class A class. So when you start metabolomic work, always think about the character of your data. Right? And also, now I will show you later on. Now is if you put 1, Brazilian drink wine often? Do you drink often? Okay, wine. Now you have tasted your wine from your market. You by your panel, you classify. Good wine is 1. Yeah, the best wine is 3. More or less good wine is 2, and bad wine is 1. Now, then is if you put 3 to 1 means actually your variable is 1. Do you understand? Because it's a taste. There is no existence of non-taste. But if I put 1, 2, 3, 1, 2, 3 is complete, it's independent. If you take independent, it's a different one. For example, 3, good wine has a own taste. And 1 is a bad taste. So it's an independent one. So depending on treatment of your data, outcome is different. So if you say 1, 2, 3 is independent one, actually you have a variable 3 variable. But if you do 1, 2, 3 as a taste label, 3 it has a higher than 1, and you have a 1 value. Do you understand? Once more, if you classify the sample, for example, sample 1, sample 2, sample 3. Yeah? Now, variable 1. Here is a mass data. Yeah? Mass data. And here is another variable, A, activity. So in the activity. So here, classification. Now, not activity is Y data, another Y data, for activity or classification. If you say is Brazilian, yeah, Brazilian, Korean, let's say American, yeah, Brazilian, Korean, American, then Brazil variable, Korean variable, American variable. Then sample 1, if it's a Brazil, 1, Korean. If this one is Korean, this one is American, like this one. So that's why if you do classification, each one is own variable. But if you say now it's activity 1, 2, 3, 3 activity, then if it's activity is activity, then 3, 2, 1. This one is related, right? Then in that case, activity 1 value. Is that clear? Then now is let's see about one example. Long time ago, I had two. I made it, not my, I made my colleague. Yeah, made transgenic plants, gene-treated, gene-modified plant, Arabidopsis. And Arabidopsis, two accession, yeah, two accession. A, B, C, D is Arabidopsis, one accession. E, F, G, H, another accession, yeah. And then each accession, I change one gene, same gene. So A is wild type of A accession. E is A type of accession. Sorry, the second type of second wild type. Then B, C, D, E, F, G, H is the transgenic plant. Now, please think about, do you understand this data matrix? That what kind of, if metabolite changes, what kind of factor do you expect? First, accession, right? A accession, E accession. And second factor is transgenic, non-wild type, right? This one. But when you start the principal component analysis, you don't know which one will be shown in principal component analysis. Why? Because principal component analysis is kind of unsupervised method. Yesterday I mentioned I brought my daughter and I gave, please separate this one. Then without any consideration, she will separate man and woman. But if I'm interested, nationality, I have to give some information to a wide data set. So please divide Brazilian, Korean, Japanese, American. You understand? So that's the power of the PLS. One of the typical supervised method is partial least scaling method. Principal component analysis, unsupervised method. As I told you, unsupervised method more or less is a PCA. There is no other options in this case. Unsupervised method, second level, to give how to provide your data, Y data, depending on this one. Every day, new method comes out statistically multiplied. So let's see. Here. Now please look at this one. That I here EFGH and the blue one is wild type. Red one is transgenic. But when you look at this one, if I want to see the difference between transgenic and non-transgenic wild type, can you see? No. Why? Because transgenic effect much lower than genetic accession effect. Right? Then I gave PLS modeling. I gave additional data. I nominated. I gave a define. This one is transgenic. This one is wild type. This one is transgenic. This one is wild type. In that case, dummy variable, because transgenic and non-transgenic does not have any relationship, which means two variables. Transgenic variable, non-transgenic variable. Now, same data set. Please have a look. Here, transgenic, and this one is wild. Now it's changing. Of course, it's not very good separation, but at least it's much better than principal component analysis. So from this one, you have to be careful. One point. The principal component analysis. Surely there is no rule. Now I want to show to separate. Then should I use principal component 1, principal component 2? Actually, no. Because you don't know your wished separation can be minor pieces. Sometimes principal component 20, sometimes 21. In that case, whatever you found in principal component combination is possible. Do you understand? Yeah. Second one. Then if you do PLS modeling, in that case, you gave already comment. Please separate based on transgenic plant, then there's a rule. You have to show this component 1, PLS component 1. Why? Because that one is already you gave a comment, then you have to use the first component. Yeah. But then if you have, okay, if you do PCA, should I check all the pieces? Because if you have 1,000 mass features or several hundred NMR features, PCA ideally can be calculated with the same number, say number minus 1, for example, 9,951. Should I combine everything? Usually, you don't have PC 1 to 3. It's not working, you can go to the supervised method. Second one is, the important one is, second one is principal component analysis. Sometimes many people, like me, draw like a circle. Yeah? A circle means actually I want to show you, but as I mentioned, basically in strict sense, principal component analysis itself is not statistical. You should not put circle because it can give a prejudice. But for to show in the paper, maybe you can draw the conclusion. Second, you don't need validation test. Why? Because you put one kind of data set, NMR, mass, UV, and then separate. There is no meaning of significance. Just to separate shows the variation. You do supervised method, validation is very important. My metabolomics data are reflecting on or influenced by transgenic or non-transgenic. You have to test this validation test. So that's why when you do PCA, no one asks you, did you do validation test? But if you do PLS modeling or another version, or PLS modeling, they will ask you, did you do validation test? That is very good. What kind of validation test? Basically, you need is the simplest one is the permutation test. Permutation. Have you heard about the permutation test?
Yes.
What's the permutation test?
When you change the classes, the original classes, and you kind of make the model understand that they're different, but actually they are not different. You just change it, swap the classes, and you can try 10, 100,000 permutations to make it significant.
If I translate the simple word, now I classified you are Brazil, you are Korean, for example. This one. And then I did it metabomic, and it's separate. Curious is my data is really influenced by Brazil, the nationality or not. In that case, there is, though, in this case, is prediction level is coming up. For example, 90% is correct. They can show. But this prediction model, absolute value, 90%, this might be, but curious is, I change it. For example, now you are original in Brazil, but on purpose, I change you are Korean, and you are Brazil. Then prediction value must be lower, like 40% or 20%. Yeah? But if my model is not correct, whatever you change, it always 90%. It's horrible. So that's why we call it it's a permutation. It's simple. Yeah. Of course, you have when you do manually, you have to change everything. But now, when I think it's a metabolic analysis, there's a permutation test. Yeah. Or simple. Yeah. Any kind of a vendor software, yeah. When you do PCA or actually PLS modeling, so there is a function of permutation test. Of course, there is a relationship or other data set, but permutation test is the biggest, the simplest. Yeah.
My validation, I can see, my test has to be able to see this little difference when it changes between us.
Yeah.
I have to see this one.
Yeah.
If I have no difference,
Then your model is wrong. And as he mentioned, so 100 or 200, which means first I change only one out of 1,000. Then from 90, prediction level it should be 89. But if I change it completely wrong, opposite, it should be 0. So that's why it's quantity. You understand? So that's how many I have to change the label. 100, more or less, sometimes 20 or 30 is possible. You understand? If you have 100 sample, you can change 100. Is that a simple version of yeah, permutation test.
Yes.
The metric values of computation
Yeah.
test. The metric values. For example, validation external values, validation internal values, and accuracy. Is metrics
Yeah.
from the models.
Yeah.
You use the metrics overall.
But ideally, when you do supervised method, my second step is actually my goal. Now I want make a model, build a model, transgenic and non-transgenic, like this one. What's the next step? I want to predict. For example, I bought another plant material, I measured mass or element. I upload it, right? I upload it. Then based on this model, they will tell you whether this one is transgenic or wild type cases. But think about this one. If permutation is completely wrong, whatever you did, Brazilian or transgenic or non-transgenic, always prediction value is the same, then the outcome you cannot trust. Yeah? For example, now you have a boyfriend, girlfriend. Yeah? What kind of a boyfriend is the best? If he is always kind to lady, yeah, of course, one time is okay. No, I don't want to marry with her, right? Only she is only happy with me, not with her. This is a very important one. He always very good character. Always, I like you, I like you, I like you. There is a problem. Always. Yeah, World War One. Very clear. This one is there. Like a kind of competition. If you don't marry, please keep in mind. Yeah? Not yet.
Goodbye.
Now, after this one, now is after this one. Yeah. Same as loading. Do you remember loading yesterday? Loading means always multivariate data analysis. You can get two kinds of data. Scope Scope means yesterday I gave an example, Bruni's family and Carol's family, and now we have original variable, height, weight, foot size. If you want to use those kinds of size, it's very difficult. After then, you reduce one PC1. That's it is. Then it's a reduced value instead of original value. Second step is, if you change the by reduced principal component 1, okay, which original value is influenced PC1, you can measure angle, right? This angle, if it's 90 degree, so original value of 90 degree and PC1, 90 degree, no correlation, nothing. But if it's a 0 degree, only height is influenced in this case. TLS modeling also is possible. Morning. And here, now it's positive one, transgenic one. Based on this one, we can draw the conclusion. These metabolites are more in transgenic. Then probably that transgenic plant is a bit touched. Yeah, flavonoid related gene, something. Now, principal component, one more thing. Now is when you do principal component analysis, you see this kind of a circle. Yeah, we call it Hoteling's 95% line. What does it mean? When you do principal component analysis, many cases you can see this line, black line. I didn't draw this one. This one means it's like when you do t-test and ANOVA test is 95% confidential now. For example, now this one is outside of 95%, which means when I consider, because t-test, ANOVA test is only one variable, right? When I consider this variable is located on in the region of 5%, 2.5, 2.5%. It's very exceptional. That's it. And this one, principal component analysis. In this model, this one is, you can say, is very exceptional. Of course, as I told you, principal component analysis is not statistics. It's data reducible. Sometimes, usually in the paper, I don't draw this hoteling slide because it does not mean anything. Yeah? But now you have data. For example, Alicia, you have several species, right? Several species. Now you measured NMR or LCMS. You did metabolomic cases. Now all the species located inside of this hoteling test, but only one species outside this one, then it's a starting point of discussion. This species, you can say, very exceptional case compared to other species. You understand? Yeah? Of course, usually principal component analysis, data reduction, you don't need it, or even I delete this, right? But as I told you, there's no perfect rules. Always, if something, you can trust it. Question?
John, if you got like one sample performing supervised learning that it's quite far. Let's see this before. And when you're modeling, you can like analyze and see that when you perform the model without this before, enhances the performance of the model. Is it correct? Can I use this strategy, or if I exclude this outlier from my original data set?
That is a good question. Yeah, this is a very good question. Now, for example, you have a model. Actually, I go to the last slide. It's actually I prepared that kind of question. Here. Now this one. Right? Now, like this one, right? And the one sample is outside. Then you are tempted. Temptation. You know temptation? Yeah? Probably if I deleted this one, probably it's nice model, right? It's it's not matter of science, it's kind of temptation. And also supervisor cannot check. Because students say, I didn't measure this one. This one, we don't know. Yeah? But before this one, person, there are strategies. Is that your question, right?
Yes.
Then in that case, should I delete or not? My strategy is, first strategy is, I don't want to delete any single sample. Because anyway, you process, whatever is processed, maybe there is some mistake process. For example, when you do normalization, before deletion, of course, at the end, there is no way to include this. Of course, you have to delete it maybe by mistake. But first one, you have to check it. Maybe because of normalization. The problem that I changed the normalization. After changing normalization from total intensity to internal standard or maximum signal, maybe if you change normalization, it can be included. Second one is scaling. Yesterday you learned this scaling. Scaling, which kind of scaling? Like unit variance scaling, non-scaling, or Pareto scaling. You change this one, which included, okay, it's one. Third one is, I'll check it. Like this one, here. He did, it's not my data, it's from outside from data. Now, this one is glucose. Yeah? And the other one is glucose, probably one spectra, this spectra, he forgot to calibrate. So now animal spectrum, glucose should be detected 5.2 ppm calibrated to 0 of TSP. But among the 100 sample, maybe he forgot, oh, one of the sample, I forgot calibration. Then this one is included, something like this. Okay? Now, then is, of course, I did it very well. I didn't make any mistake. Normalization, so the normalization, calibration, or the scaling, no problem. But then this still is too early to delete the data. In that case, you have to go to the supervised method after PLS modeling, after OPLS modeling, or independent component analysis. When you change the Y data set, probably those exceptional data is included, like my case. Yeah. 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 Something, yes, a PCA does not have P4 is included, but when you change to this one, yeah, here P4 slightly included, but when you do OPLS daily, it can be included. So deletion is a really last step. And also, when you want to do, sometimes here, now this data has a two factor, transgenic and accession. They are different one. When I am giving a Y data set transgenic, which means I will blunt. Now I don't want to look at accession cases to suppress maximum variation. So in that case, now it's a simple way, it's PCA. Some people did it. Okay, then I take out because from my principal component analysis data, A accession, B accession is a two major. Is a two different. Then I will do PCA only separated A accession and B accession. That is also possible. But for me, first I want to include it for PLS modeling, I suppress oxidation one, then you can always at the end, statistical model is significance is based on number of samples. So that's why if you delete it, your number of samples always is very low. And also when your case is number is always small. If you divide only half, then it's very insignificant. So that's why to delete or to divide, including divide is the last step.
When I use normalization scaling, we are talking about comparison, as I said, between sample, need. But in the practice, if I have those comparisons depends on repeatability, relevance of my analytical test. For example, in LCMS, if I have 100 samples to analyze, when I think about repeatability, considering the sampling, preparation of my sample, the equipment. What is better? Analyze 100 samples, or, for example, each 20 samples, I check the calibration of
You can ask Peter to answer. There must be an answer. It depending on the instrument. Animal cases, for example, now you have 1, 2, 3, 4, 5, 6, 7. You can name it, right? Number, sample code 1, 2, 3, 4, 100. But number 1 to 10 is a transgenic. 11 to 20 is a wild type. Now you change it, you separate it, transgenic and the wild type. Then there are two reasons. It's because of genes. The second one is because of order of sample, right? Because who knows in the beginning of 10 sample, temperature is stable, next sample is non-stable. Animal cases, I don't think you can find that kind of technical variation. If there is a variation, you can see animals characterized. Sometimes wrong swimming or wrong everything you can find. But else in this case, there are too many signals comes out because first one is random sampling, not transgenic, not wild type. Instead, it's mixed up, random sampling. And second one, as I mentioned in the beginning, you always estimate GC given QC sample. Every 10 sample, you measure the quality control sample. For example, quality control sample is the very easiest way. You mix up all 10 sample, the same amount, you extract it. Always check every 10, 11, 21, for example, and 31 is a QC sample. When you do PCA, all these QC sample is centered, sorry, is in the one location. If QC sample exactly same sample somewhere, this one, then your data is not trust. So mass scale, LC-mass special. Any objection? Objection? Objection, Alithia? No. Now, there is a variation of a principal component. It's not PS modeling. Variation is a multi-dimensional scaling or single value decomposition. Have you heard about this one? This one is kind of alternative or a variation of principal component analysis. Like Alicia cases, if we have a very small number of sample, those numbers, sample number is very small. To solve this one, maybe you can use single value decomposition method. It's kind of PCA, but my opinion is you don't have to use. Nobody is using. Nowadays, PCA, you can have another method to read. And this one is quite useful. Multi-dimensional scaling efficient for non-linear projection, which means when you do cluster, Brownies family and the Harris family, we draw the first line, right? First line, this one. To get maximum variation, to test center, right? Instead of this one, if sample is 2, cluster 2, disperse, maybe second order line or third order line you can draw. But as you know, any kind of instrument, any kind of method, always default is the best. One of the tips to give you, you are starting new project or new technique. Yeah, then you have to click. Yeah, then please choose the first one. That's the best. Yeah? Always there are some several methods. You don't know the first one. This one is second or third choice. But if you want to apply, another problem is if you want to apply non-orthodox method, right? There's a variation method. Always you have a clear explanation or rationale. You have to explain why you have to use this one. Well, at least you have your data should be increased, improved. Second example now is
Try. Before you start in the example, just one question. Is appropriate to use the PCA when you're working with a data matrix that you have like a lot of absence, like a lot of zeros? So like you have more zeros than actually values. Is it all right to use the PCA or is something wrong with this? Because
any answer? It's a very difficult question. I have some tips, but do you have any answer? Animal case, many cases, sometimes if you selected the signal, selected the signal here, now pick one. Everything is selected, but those two are 0, right? And this pick, I selected the variable. Oh, those 0. There are too many 0, right? If you have a 0, first one is data significance is getting down. How much is tolerance? More or less, they say 30%. More or less 30%. 30% is acceptable, but this one should be confirmed by significance. Significant. So then there is a rule, right? Rule how to fill the empty value, because always when you do multivariate data analysis, Zero is a value. Zero is a value, which means not detected, but missing value. We call it missing value, right? Missing value is around 30% is acceptable. But to avoid, if you have too much, there are several ways to fill the missing value. What's the way? Average. Average one. Median value I add this. Average median value. Or if you give this one, then there are several ways you can invent it, but one of the easiest way is now this missing value, then I calculate median value, I add median value. Then which means if you have a median value, probably the security is secure, your PCA data, it does not affect a lot. Yeah, but if it's not normal distribution, you better use median value. Yeah? Then you will lose the importance. If it's really there is, if there is a signal there, but by mistake it's deleted. If you use median or average value, yeah, mean value for this value, you will lose the effect of this one. Right? But NMR cases usually any one there is a value, but mass cases, there are many zero value comes out. Yeah, zero value is missing values comes out. But you have to think about it. Now, I did it. It's one of the quite successful results. I published it in 2006, and which means I started the project 2004. It's around 20 years ago. Still is cited a lot. It's very well designed experiment. Now, tobacco. You know tobacco? Cigarette sample. And one of the famous model is tobacco mosaic virus. I decided to this one because this one is not replicate. When you extract plant material, there is no metabolized from virus. This is easy. But if you do bacteria or fungi, they are growing there. Then when you extract it, if you separate the two plant material, maybe the difference because of the infected one contains more bacteria or more fungi cases, but virus is okay. Yeah? Now, let's say, when you do two plant material, I treated here tobacco mosaic virus. And then this one is a control plant material. And then this one is directly infected one. This one is upper lips. We call it distal lips or systemic lips. Yeah? Probably, if infected, the defense mechanism of plant shut on, and then maybe some metabolites is coming out. Now, when you look at this data, if you compare this one to this one, now, for example, it's changed. As I mentioned, this one is young lips, this one is older lips. We don't know whether this one is because of age, whether this one is because of infection. Something like this one. So this one must be here. This one must be here. This is a very important one. Second one is when you do plant material or some infection disease, you need a time frame, right? After infection, sometimes there is no symptom, even biologically. Usually it's a few seconds. In metabolic level, there is no symptom. But you need a time, 1 day, 3 days, 7 days, then 10 days, up to 2 weeks. Usually in the beginning, within 1 day, primary metabolites are changed, and secondary metabolites is going to up to 2 weeks. So now, when you find the metabolic difference, these two groups, what kind of factor? Always you start the project, you have to make a list. First factor is infection, right? Infection. Second factor is developmental stage of and the old and young, more developed, less developed, and also age, 0 day after infection and 10 days. 3 factors are included. Right? So, but depend now is a 3 factor. Actually, I want to know about the infection itself, but uncontrolled one, an uncontrolled factor, but defined culture called the developmental stage plus age. Of course, 10 days, it doesn't matter for you. Yeah, 10 days ago, your photo. Now this photo is the same, but for plant cases, it's like 10 years, completely different. Now, when you do here, all sample ideally PCA. Good. Looks like some changes. Well, green was the lower leaves, sorry, upper leaves, and lower leaves, lower leaves. Yep. Upper leaves, lower leaves, and also upper leaves, lower leaves. For example, now maximum variation is the biggest variation is not infection. I want to see, but I cannot see. Only this, because it's this and this. Alicia, now you realize, when you collect the plant material, one day you collect the upper leaves, one day you collect the lower leaves. Probably it's because of developmental stage, young leaves, older leaves. That's the biggest thing. So now, as he mentioned, as mentioned, now I want to see only local leaves and lower leaves. Then, which means I only take out, take out. I didn't take out local leaves. So local leaves and locally infected leaves, these leaves, and now healthy leaves here. Now it's separated. Right? Separated, but 1 day, 3 days, 7 days, 10 days. The not best, the typical variation by infection, always I claim, is a V formation. V formation means in the beginning, the effect it didn't do, it is still silent. Yeah, so that's why in the 1 day, more or less similar, right? And then 3 days, little bit bigger, and then it's bigger. V formation, local leaves. And systemic leaves also same. That is only I did it with the PCA. But now, as I mentioned, I don't want to take out the sample. I want sample existent. So I did it PLS modeling, PLS DA, discriminant analysis, by what? Infected, non-infected. I don't care about age. Yeah, age from 1 day till 10 days. I don't care about developmental stage. Lower leaves, upper leaves. I give a comment to my daughter, to PCA, PLS modeling. Would you show me whether it's infected plant or non-infected one? 2 classes, yeah? 2 classes. Please look at this one. Very well separated. Now you can see, and which metabolites infected here? 5-O-caffeoylquinic acid and nicotine 4-caffeoylquinic acid. Something like this one is you can find. So, which means it's to your question, should I delete the sample or not? It's too early. You can do PLS modeling existent. If you separate this one, why we have to delete the sample? With a very long excuse. You can do this. Second one is, I'm not interested, same data set without any sample deletion. I gave a comment. I'm not interested in infection. I'm not interested in the developmental stage. But I'm interested in age effect. 1 day, 3 days, 7 days, and 10 days. Now, let's look at this one. 1 day, 1 day, 3 days, 7 days, 10 days. Now it's PC1, 2 is not enough. I do PC3. Yeah, here, 1 day. Now there's effect. But then you can see here, something like this one. But this one, I did it 20 years ago. I have a question actually to myself. Now, if you put PLSD 1 day, 3 days, 7 days, 10 days as an independent variable, which means biochemistry 1 day metabolism, 3 days metabolism, 7 days metabolism, 10 days metabolism, it's independent. But that is actually not one. That which means 1 day, 3 days, 7 days, 10 days. I treated this one is like a nationality, which means 1 day, 10 days, there is no correlation, only different concept. But instead of this one, if you give an age effect, 1, 3, 7, 10, you have only variable. What kind of, now this one is 1 day, 3 days, 7 days, yeah? Sorry, it's 10 days. 10 days, yeah? 10 days effect, yeah? Then which means I treated this one. But as it is, it's not very understandable. Instead, if I give only one variable, age, then you put 1, 3, 7, 10, then which means getting older, which metabolites is higher. Getting younger, which metabolites higher. Then you can get these cases. Do you understand? So if you have even numeric data, you have two kind of choice. Would you treat them as a text? Would you treat them as a number? This case is I treat them as a text, which doesn't make any sense. Instead, we got to use, actually, it's naturally age is age effect. Question? So message is technique, but always when you start the project, you have to investigate the factor very carefully. And always I'm asking students, when you have a factor and response, please make a list in the Excel file.
Choi, can you just go back the slide one for this one? One more. Just a doubt. Can I say, or I can't, let me reformulate. Can I say that PC1 is able to separate local and systematic leaves based on the distance of the variance that I'm seeing, visual seeing there?
No, but
or not?
No. Why? Because if you say, okay, it's a systemic leaves of a systemic leaves. What's the systemic? A systemic leaves and local leaves, right?
Yes.
It's separated. But in that case, hybrid plants also upper leaves, lower leaves is changing, which means now you found this difference, right?
Right.
But you can find also this difference, which means we are not sure whether this one is because of systemic infection effect or is because of developmental stage effect. This is a doubt, right?
Good.
So then in that case, you need a PLS model.
Thank you. Good point.
So once more, when you find the result is result, you have to think about this. Otherwise, as he mentioned, okay, now I found the local and systemic this local and systemic is separated. Okay, in PCA, I found it. Then immediately someone asks you, how about this upper and lower limits? It's changing. How can you explain this? What kind of difference? This is right. Always, not automatically. When you from now on, you will use a lot of computer or web-based network or modeling, whatever. Always, when you get the data, please, 1 day or 2 days, please look at your data by yourself. Is it possible? Or something? Please ask yourself. Now, is there any difference between local and systemic leaves? Yeah, look at this one. Now, here, lower leaves, upper leaves, right? Lower leaves, upper leaves. Here, this one, systemic leaves, and every fork is coming here. Every fork coming here. Now you can find which metabolites higher. In this case, we can find very nicely working. So what's the relationship between metabolites? When you do loading plot, for example, loading plot, now here, when you do, loading plot, cosine alpha, then if two compounds has the same effect, so now let's translate to apply to the another application. Now you found it, you tested it, your plants, these are some metabolites, so metabolomic alchemist. You treated anti-inflammation data. yeah, anti-inflammation data. Now, based on loading plot, you found the two compounds or three compounds as an effect. Then it's a starting point to look into another possibility, synergism. Right? Two compounds, okay, related to this one. Now, two compounds, right? Activity, increase activity. Now, here, capsidiol and sembrane, two compounds. Those probably is behave in a similar manner. One possibility, of course, it cannot be synergism. And also, some people say, as he mentioned yesterday, 2 days ago, he showed. Now it's based on correlation data, based on multiple data analysis. These two compounds, alkaloids, related to correlated to activity, as he got a conclusion. Yeah, maybe. Then, as it's in strict sense, it's not necessary to be those two compounds are active. It's just a correlation case. Who knows? Some compound which is synthesized after this alkaloid really is active, but LCMS only can detect, NMR only can detect this compound is correlated. So that's why if you want to draw a conclusion based on metabolic correlation or pairs modeling, I found that this metabolite is a candidate for this activity, but you have to prove it. I isolate the compound, or I own the reference compound, I test it. It's really nice, something like that. That's the sense. Strict sense, you have to say it's a correlated activity, correlated. Also, another reason is maybe these compounds can be synergistic. So, metabolic analysis data, metabolomic analysis data, yeah, is not end. It's always can be starting point for another research. Second one is, if these metabolites, then you might have a question. If we want proof the activity, I have to isolate the compound. Then why I have to do metabolomic work with the LCMS work collection this data? It's completely different story. If you start unknown plant material without any information, it's really takes a really long time, right? 10 years, 20 years, because we don't know. But now, based on iris data, we found it. Okay, these two compounds related to activity, then you can now targeting, isolate this compound. It saves a lot of time. This is a different story. There is a meaning. Or if it's not possible to isolate this compound. Why? Because it's not commercially available. Even when you isolate this compound, the amount is picomole, then you can maybe invent new approach, like a strategy. For example, I ask my colleague to synthesize. If it's difficult, maybe then I can find some commercially available compound which has a similar structures and structure activity relations, something like that. What? Molecular docking system. Have you heard about the canonical correlation? Canonical correlation is another story, but canonical correlation is not supported by metabolic analysis. It's not often used. After data reduction, so correlation between the sample. Simply speaking, here. Now you measure the honey sample. Yeah. So honey means honey product is here. And then around the bee hive, right? Bee hives, you can measure IR animal mass. I measured honey, which produce certain region. And I measured IR spectrum, or animal spectrum of all the plants, like orange blossom, carrot, wildflower, whatever. After that, there are two kinds of data sets. You give a clear which one is correlated. This honey is related to carrot. This honey is related to this carrot. Why? Because this one is in honey, there are two factors. The honey is influenced by another metabolome of plant one. Yeah? Two metabolomic data, honey and plant, you correlate this one, you can classify based on this case. But this canonical analysis is very difficult to find some software nowadays, free software, because you don't have to do. Now, if you do OPLS or PLS modeling or hierarchical clustering analysis, the variation you can use it, but in all the time we use this canonical clustering analysis. Now, cachaças. Yeah, you measure all the cachaças. And then around there, you measure all the sugar canes. Yeah? Then based on sugar cane, which sugar cane used for which cachaças. That's the solution. And cachaças, this case it actually is not a good example because any kind of cachaças use sugar cane. Which sugar cane does not have does have very little variation. Maybe another additional plants we need to do. Like genuine, fake. But in some plants, it's from here. It's from sugar, then this one is a fake. You understand? And PCA, I told you, after raw data, principal component analysis is an unsupervised method. You must do, right? After PCA, of course, you can do all the supervised method, PLS modeling, OPLS modeling, something like this. Another reason is, please look at this one. Another reason is, there is rules, the ratio between the sample and class and variable. Now, many people want to, because you have already LCMS, but to collect the sample, it takes money and it takes money. In the beginning, Gilberto mentioned, now I measured only one sample, two samples, three samples. I detected thousands of metabolites. I want to classify based on three samples. It doesn't make any sense, right? You need a replicate. Then there is a thumb rules here. Number of variables should be less than number of sample minus category class. If you put 10 category or 20 category, which means you need a more number of samples. Now, if you have only three classes, NMR spectra 256, you need at least 80 samples. Can you imagine? Now, if you increase the category a lot, then I think you increase 200 samples, 300 samples. Of course, nobody is following this rule. But now, then what's our solution? I have a fancy method, LCMS, NMS, but I don't have a colleague to go in the middle of Amazon because only 3 samples, 10 samples. Yeah? But then question is, easiest way, you have to reduce variable. Reduce variable means LCMS, I will do targeted analysis. Only I detected one compound, two compounds. It's a big deal. Yeah? In that case, now please don't forget principal component analysis, data reduction. Now you have 1,000 LCMS data, you reduced PC1 and 2, right? So instead of 1,000 variables, you can use PC1 and 2. Then you can handle this kind of data. So after PC, you reduce the data. Then each sample, instead of height, foot size, or weight, instead of this one PC1 score, right? Reduce this PC1, PC2 score, you use for further canonical analysis, independent component analysis, or hierarchical cluster analysis. You can use it instead of original variable. So PC, there are two purposes. By reduction, you can see classification. Second one is you reduce data, this reduced value can be used for other sophisticated analysis. Don't forget, this one is sometimes, of course, hierarchical clustering analysis. Like sometimes for the classification, you are using the phylogenetic trees, right? You can use original mass data, but sometimes significance is very low. In that case, please use PC, then you can do the classification. Yeah? Then based on small number of samples, you don't have you have a very clear excuse. Look, I have a limitation to collect all the species. I have the richness animal, thousands of samples. I cannot have a more samples. Instead, this one, I reduce the species 1 to 3 instead of 1,000 samples. I get this phylogenetic tree. This one is very good.
And try. You also can use the PCA to check the quality assurance of your data processing, as you mentioned, to seeing the quality and the replicates.
Yeah.
So it's also used for that.
For example, yeah, this is a good question. PCA also is there are many applications. Now you have a triplicate, also QC sample. Yeah. When you do this one, if it's a triplicate, sometimes even animal I add it one sample. I don't have to measure all the sample. Technical triplicate, biological triplicate, fine. But technical I did. But one time I add it. Yeah. 10 kinds of sample. I don't have to measure each sample technical technical application animal. But one time I choose it. For example, number 7, triplicate. Ideally, this 7-1, 7-2, 7-3 should be on the same spot because it's a technical application. If it's spread out, then your method is problem or your machine has some problem. That is, I think it's your question, right?
Yes, sir.
So we have some minutes, right?
Go ahead.
And
My
now,
mother has class only 11 for
no, it's not. Have you heard about the Simca? Others, you heard about the Simca, right? Simca is a name of the software, right? Name of software. But there is a model. Actually, Simca is independent modeling of class analog. I told you, nowadays many methods they use the jargon, yeah, which we don't understand. But question is, it's simple. It's for the classification. Alisa, it's for you actually. Now you have 45 species also, yeah. Alves, you also say you have 100 species. You measured LCMS NMR. So first, your idea is you want classify, right? Which species close which one, right? Which species is clear? Then you can do after PCA reduction. What original data you can do? Phylogenetic tree, they are close. This species close. Another one is if we have a target species. For example, now this one is here. For example, it's a Korean ginseng. Chinese ginseng, Vietnamese ginseng, European ginseng. After PCA model, only this one, each, yeah, Korean ginseng, Chinese ginseng, and what's that, the American ginseng, Vietnam ginseng, I made distance. Yeah. So now you have a class sample. But this one is you need a more number of samples. But in case now is you have a 10 sample of Korean ginseng and Brazilian ginseng and American ginseng. You accumulate Korean ginseng they said is the best quality. Then you want to know the Brazilian ginseng, American ginseng, which one is close to Korean ginseng? This is a good question, right? Then in that case, you have a model of PCA. PCA based on this one, the Korean ginseng. You measure distance. Brazilian ginseng is closer than American ginseng. Then Brazilian ginseng is close. Now, Alisa, you have a 45, for example, very limited number of samples. But sometimes you have some species, you have a lot of number, right? Some species. And some species only 1 or 2, right? In that case, okay, if you do phylogenetic trees, they said, oh, yeah, the number is not well balanced. Some of them a lot, some of them is small, right? In that case, maybe you can change the research for. I have a model, this sample which has enough number of sample, that other sample, whether they are, which one is close or not. That is also possible. This is a soft independent clustering model analog. You understand? So difference is hierarchical clustering analysis is a classification of all. But Simca modeling is from standpoint which one is close. So now here all the species here, then hierarchical clustering analysis or phylogenetic tree, we are correlated distance. But Simca modeling is we made a model. We are Korean ginseng. Yeah? Then Brazilian ginseng or American ginseng distance. Even that compared group does not have to have a more numbers. Only his model should be higher. Yeah? This one is nowadays, even now, I'm often used for the classification. Example. Now it's a final example. Example is this is like a mate experiment. A mate experiment. Here now is application to chemotaxonomy elect species. I think I published it around. I'm getting old. I think it's 15 years ago. Yeah, I mentioned here elect species. You know mate. Who is drinking mate? Yeah. Mate is Ilex brevicus, Ilex tumosa, tejans, all these species. But among those one, our ancestor Paraguayan Indian, they chose Ilex paraguayensis. It's a drinker. It's quite interesting because at that time they didn't have NMR mass. Yeah? But all these 20 or 10, 11 species is autonomous. How is it possible for American Indians select less than Paraguayans? It's only refreshing beverage. Then I collected this plant material, this one. The first question is, why Lex paraguayensis is so different from others? That is the first question is. Second one is, now I want to know which species close, which species is farther. Maybe which species is very close to Lex paraguayensis. Maybe it can be alternative market, something like that. So for this one, now it's kind of a model. This one is a lucky case because I'm not big traveler. My friend in Argentina, an herbarium guy, he traveled all over the South America, here, Brazil, Paraguay, Argentina. Since 1989 till 1988, he collected all the seeds. And then he grew in his garden, but it's not second generation, it's the first generation. Even then, it's okay. To avoid this one, same species we collected different year, because who knows, so it's different. Same species we collected different place, like from Paraguay and from Brazil. Then we try to remove all the environmental factors, because seasonal variation or geographical variation. Two variations we excluded. And practicum. And we did it, animal. At the time, animal looks very nice, with species. But now, here, for example, now we changed it a lot, all the species, in this case, research question is very important. Now, when I measure the Hessianus or animal, 11 species. Then my conclusion is, after this one, each species has its own cluster. Is it interesting? Answer. Of course, it should be different. Yeah, if you collect a different plant material, it should be different, right? It's not surprising at all. So that's why this kind of research, you have to be very nice formulating the question. Okay, now it's fun. After that, now we have some groups of metabolites. After that, this one. Now species 8, Ilex paraguayensis. My first question, paravolensis is really different from others. Now, let's look at this one. Very different. And the other one is 9 is a little bit close, but still is a completely different species. Now, conclusion is our ancestors in Paraguay, they are perfect. Yeah, they are perfect. Nice. Collect this sample. After then, we did some 2D NMR, or which kind of metabolite it is. Please look at this. Relax Paraguayensis, heat map, yeah, annual spectrum, and to most of whatever species. Only those 1, 2, 3, 4 compounds have, as expected, only Relax Paraguayensis contains caffeine. The other one does not have any picomole of caffeine. It's strange, yeah? It's surprising. Now, this looks here, very different this one. Then, question is, now, here, maybe 9, species 9, yeah, species 9, or other species, probably, if now is, I did it, but I didn't publish this one. Maybe I want to develop a natural decaffeinated, yeah, decaffeinated marker. This is nice. For example, now I take out the caffeine signal, right?
Yeah.
And then each one shows the best, yeah, similarity to matter. Then I can claim, okay, let's Dumosa probably is the best profile, but no caffeine. It's a sales, marketing sales is a target change, something like this. Always, while you are doing, you have to formulate your own research question. Yeah. Now it's, yeah. So probably you realize that the metabolomics now is more or less is going to the conclusion, right? See? So nothing new. Yeah, nothing new. I already showed, yeah, except that you use different direction. What kind of direction you have? You want, see, old. And combination of present technology. I'm not interested in animal. I'm not interested in SMS. I'm interested beyond technique. And third one is integration of many research fields. Yeah, metabolomy. Someone asked me, is there any textbook to learn metabolomy cases? But it's rather difficult. As you see, you have to read biology book. You have to read natural product chemistry. You have to read statistics. You have to read analytical chemistry. You have to read NMR. Something like that. Then, which book is the best? I would say there is some metabolomics books. I sent her some books, titled to Fatima. Maybe she can relate to this one. One of the protocol book, which is written around 2009, Wolfram, Wolfram in Humana Press. That's when you are studying that book, then all the protocols are included. That is nice. Then, if you want to know, analytical chemistry, you want to learn NMR and SMS, right? In that case, still, when I'm giving a lecture, always I prefer Silverstein book. You know Silverstein? Yeah. Silverstein book because it's included NMR. Now, many people say, oh, I read it. One time reading is not enough. It's not enough. Every time you read it, even now, I found a new phenomena. Even all the interpretation written in Silverstein is nice too. Yeah? And also, of course, you can use a Pavia book, it's equivalent of this, but that and also mass cases, one of the big points in mass spectrum, when you SMS metabolomy, we are now is modern mass scientists or metabology. They are very good at computer algorithm or to take a data. But now, future is we need a fragmentation. Based on fragmentation, you have to structure and elucidate. When I learn the mass spectrum, always based on NMR spectrum, without mass spectrum, without help of NMR, I can draw the structures. So like M-18 is a water signal, M-16 amide signal. You have to start with this. Then mass fragmentation. If you want to learn mass fragmentation, which book is best? You should not read the colored book, it's a black left book. The red covered book is still very nice because it's based on EI spectrum, but he described very well all the fragmentation pattern. So now sometimes you found a very weird fragmentation cases. This one is with the magnetic.
You have the different phases from
Of course, depending on different ions, fragmentation is completely different, but EI fragmentation includes everything. If EI spectrum is 100, ESI shows some part of EI. O track some part of this data. And also, when you do simulation, now you can do simulation, the fragmentation cases, the like Excalibur, long time ago, those kind of things based on EI stuff. Statistic, of course, you follow the statistic. I don't want to introduction. Multivariate data analysis. Which book is the best? Metrics book, metrics multi and mega data analysis. Which one is it? I can show you. Yeah, this one. Even it's a 2006, but there is a revisions there. Multi and mega data analysis. This one is, for me, is very easy. It includes PCA. It includes PLS, OPLS, multi-regression, SIMCA modeling. Everything is there. This one is not free, of course. If you don't have money, Chapter 3 is available for free. That one is the basic of PCA, which is very important. And if you want to know, now is some people, their background is not phytochemistry. You want to know which kind of compounds related to plants or not. In that case, now if you want to do pharmacognosy or natural product chemistry, you cannot find the proper book. It's very deep, right? In that case, the Swedish the pharmacognosy textbook, drug development from natural origin. First is a deep. First, all the information of biosynthetic pathway and structures are included. What kind of flavonoids are included? What kind of saponins are included? What's the definition of this one? Drug development from natural origin from Swedish is Umeå University, Uppsala University textbook. This is very nice book, very thin. Any questions? Any textbook do you need? So here, now this one, and conclusion, PCA for this conclusion, PCA is unsupervised method. After PCA, you can do PLS, and then now OPLS. I didn't mention OPLS, you can read it, this case, but it's similar as OPLS. And DA means same, but you are using qualified class data. After PCA, you can go to SIMCA, canonical analysis, independent component analysis, after reduction of data. This kind of data, sometimes you need a limited number of variables. Sometimes in computer it's not working. In that case, after reduce PCA, you can use it. You heard about the factor analysis. Old version of statistics is very similar to principal component analysis, but philosophically it's very different. Like Greek philosopher, like Socrates, Plato. He always based on this one. He wants to know the harm of his work. Factor analysis, PCA analysis, factor analysis, data look like factor 1, factor 2, PC1, PC2, same, but principal component analysis, data reduction. I showed you yesterday, one X, Y, Z, put this one. Factor analysis actually is started from the psychology. For example, Einstein and Mozart, both are genius, right? Einstein's genius expressed in physics and mathematics. Mozart's genius expressed in music. But we interested, what's the core factor? What's the core factor of geniusity? Based on this expressing, it's like a Greek philosophy, like Plato, or Parmenides, something like that one. They are focused this one. So that's a principal component. When you add in 10,000 or 1 million data, you can reduce it. No problem. It takes a bit time. But vector analysis, when you want to reduce it, because they want to look for others, it's very difficult to calculate. Sometimes it's not very well calculated because it's a kind of philosophical model. So when you find some analysis, vector analysis, when you get it this one, probably it's easy to do it, but calculation is difficult. Nowadays, it's very difficult using vector analysis. If you have more than 200 samples, you cannot use vector analysis. But it's just for your reference. If you meet vector analysis, it looks like a 1980, 1990 paper. It's very similar, but philosophy is different. Here, I already showed this one. Yeah, if you want to delete it, if you want to delete some data, yeah, to show off your result, there are several steps you have to follow. Yeah, and keep in mind, it's like metabolomic is not a goal, but tool. Yeah, you should not be proud you learned metabolomic. You should be proud you applied metabolomic to your research question, and studying metabolomic is not enough. Or yeah, in 1970, many people say, "What's your major?" I'm doing gas chromatography. My major is gas cross GC. My major is HPLC. Now nobody is saying like that, like this one. Metabolomic. We study with metabolomic is very important, and biological or engineering question, scientific question. Let's say scientific question is very important. If you have wrong scientific question, you are barking for wrong tree. And another one is, this conclusion is still our technique can detect, identify very small percentage of metabolomic. I told you in a single orange, 30,000 metabolomics, 30,000 metabolic, but we can detect maybe 100, 150. It's almost nothing. To solve this one, yeah, maybe we can use this kind of a biosynthetic pathway. Why? Because black one is I cannot detect, but colored one I detect. Probably then you have some story what happens there. Second one is our current metabolomic is a snapshot, just a snapshot. There's no information time. There is no information space. For time, you have to use flux analysis, fluxomics. When you feed sample, how they behave. Of course, it's very difficult to go to the secondary metabolomics, but primary metabolomics, many people are doing fluxomics, yeah, fluxomics. And second one is space. You have to consider now, if someone is doing image analysis, particularly human metabolomics. I have some liver, mal liver function. Then you can take the tissue, the targeted analysis. Plants infected. Then this part, which metabolites? It's interesting. So space still is missing point in current metabology, and time also still missing point in metabology. Any question? Exactly 10 o'clock. Yeah? Sorry?
Some question.
Yeah.
User email. She asked, I could not understand when I use a thing as MGS and SVD. Could you explain again?
SVD.
Yes. MGS.
Is this one, right? In this case, it's the variation of a principal component analysis. First one is your sample, number of sample. It's very small. Yeah, but I want to do proper correlation, proper separation. In that case, you can use singular value decomposition, SVD. But second one is, if your data, for example, glucose level does not follow first objection, you want to draw the projected line, not first order line, second order line, or third order line, then you can use this one. But before this basis, nowadays, very rare people are using this method. So she can forget about it. Second one is, if you want to apply any of them, you have a very clear explanation. First one is, especially first one. Why do you don't want to use the first order line? This is okay, but it's very difficult because natural variation. Sometimes when you look, you don't know whether it's following the normal distribution or not. Yeah? This is very difficult. So most instead, my suggestion is, if you do PCA is not enough, then you can go to the supervised method like PLS, OPLS, SINCA, whatever. And or best way is please increase the number of samples. I hope Lucia thinks. Any other questions, guys?
You talked about this book of pharmacology from Uppsala University. Describes description about the main class of pathology that you can found in different plants. I don't remember the name of the author or the protocol.
Which one?
What is the title of this book from Uppsala University?
Drug development, I vaguely remember, drug development of natural origin. Drug development natural origin. Please find the books. Because most of natural product chemistry is a very specialized, very specific topic, or it's too deep. But this one is a proper volume, and include a very diverse topic. There is a Japanese book, but I don't think you can read the Japanese. And when I learn the natural product chemistry, of course you don't have to do right now. Because at that time there are not many books about this one. I wrote one Sigma catalog. Nowadays Sigma catalog online. At the time they are published the book. But then when I don't have to remember all the name of the compound. At least when I check the structure, I will exercise myself whether this one is natural product or synthetic compounds. That's the first step. So when I show the structure, you have to tell, is this one is natural product or is this one is synthetic compounds? That's the feeling is very important. So you can read all the book, because you can remember, you have to show off your knowledge. But before that one, when I show your structure, first question is, can you tell me this one is synthetic or this one is natural compound? This is the first question. And statistics, the statistics comment is statistical minimums that ANOVA, even ANOVA or whatever t-test or MANOVA, always, unless you want to be a statistician, you don't have to test this one. Instead, always you provide or you make a nice list, factor and response. Response you can do with a statistician. What kind of spectrum, what kind of data analysis is good for them? They will validate it. Any further questions?
Why is that you treated your data as a text rather than numbers? What did you mean?
Text means, now you have a 1. I'm 2, for example. If you treat the text 1 and 2 is different, like you're Brazilian and Korean. 1 is Brazilian, 1 is Korean. But if you treat it 1, 2 as a number, I 2 times bigger than you. Okay? Then if I treat the text variable 1, 2, you have a 2 variable. Your case is Korean 0, Brazil 1. My case, Brazil 0, Korean 1. But if we treat the number, it's only one variable, number variable. You 1, I'm 2, the other one 1, 3. Okay? That's the best, I made a bit simple version. Don't drink too much tonight. Other questions? Because today is probably someone is my last time to meet you, so if you have a question, so feel free. Thank you very much for your attention. Thank you very much for your interest. I'm really happy with you, yeah, even more than I expected. So hopefully next time we will meet in the same holiday. I'm sure we'll have a nice future. Thank you.
Thanks a lot, Joy. Your course was excellent. I'm sure the students could learn a lot with you. And I think the student could learn exactly how to think about the project. It was excellent. You could show that the student need to think about the project. Thanks a lot. I remember that at 11 we have a conference from Professor Vanderland. Please, I hope I will meet you again 11 o'clock. Okay? For you. Thanks a lot. Marlon, we have a lecture from Professor Choi so that we can see some examples about his research.
Last but not least. Yeah.
Quer que eu fale? Porque eu disse,